Hi, this is Eric Lyons, and I'm going to give a quick tutorial on using organism view inside of CoGe for quickly generating syntenic dot plots between some genome of interest and something that's related to it. So to begin, you can search for an organism using organism view. And let's say I'm just curious and want to explore some bacterial genomes. So I can begin by doing a taxonomic search. This is on organism description by typing in bacteria. And now most of the genomes in CoGe have the NCBI taxonomic description, so you can find everything based on these terms. So this is a quick search for everything that matches bacteria, and here's the example of the taxonomic description. So I can scroll through here and see that there's a bunch of acetobacters, and what I want to do is pick one of them for comparison, and I've selected this one, and its information for the organism is displayed up here. And there are now links inside of this panel that will take you out to Google, for example, if you wanted to find some more information on what this strain is, as well as lets you quickly modify that search. And likewise, you can go out to Wikipedia. Often you have to edit it just because there's additional information that's associated with those names. And once I've found a genome of interest, I can click this link under genome information, which will send me straight over to SynMap. Now when SynMap loads, both organisms that we're going to compare have been specified based on what we found in organism view. But what I want to do is I want to find one that's related to it. And so now there's links in the taxonomic description, so if I select Acetobacter, it will then find all the organisms that match Acetobacter, and I can pick a different strain. So we'll go for this strain right down here. Oh, that one just contains a plasmid. So you can quickly scroll through and find one that might look interesting. So we'll take a look at this one. So once they've been selected, you just have to click the button Generate SynMap. Now these genomes I haven't actually analyzed previously, so it's going to take it a little while to generate the FASTA files that are then blasted against each other to search for putative homologous gene pairs. Now those putative homologous gene pairs are what's used in order to identify synthetic regions through collinear arrangement of genes. So this is if you see genes A, B, C, and D in one organism and you see them in the same order in the other organism, they'll light up. So now here's our synthetic dot plot. We can see that for the most part these two genomes are identical to one another. The green lines represent the synthetic gene pairs that were identified, and has a couple of plasmids and looks like some repeat sequences that are causing some of these patches here. So thank you, and I hope this tutorial was useful.